Dear student, good morning. Our lecture today is about Humoral Immunity Part II. We will discuss some items and discuss something we didn't went through in the last lecture. Now, here, let us discuss something about anatomy and location of the immune system. A fundamental difference between the immune system and other body systems such as nervous system, endocrine system, and other body systems such as digestive system also is that many of the cells involved in the immune response are highly motile. They use the blood vessels and lymphatic vessels in order to move into and out of organized lymphoid tissues and to reach the site of an infection. Now, for effective acquired immune response, an intricate series of cellular events must occur. First of all, antigen must be detected and then detected and processed by antigen presenting cell, which subsequently make contact with and activate T helper cells to stimulate B cells in that case, and cytotoxic T cells precursors. In addition to the cytokines also that are required to support lymphocyte proliferation. Secondly, memory cells for secondary responses must be formed and the whole response coordinated to that infection. It is adequate to that infection, but not excessive and appropriate to that infection. Now, what about the antigen being dealt with? Need integration of the complex cellular interactions that form the basis of the immune response takes place within the secondary lymphoid tissue. First of all, the primary lymphoid organ, such as thymus and bone marrow, while secondary lymphoid organs are lymph nodes, spleen, mucosa-associated lymphoid tissue, mouth, in the lining of respiratory, gastrointestinal, and genitourinary tracts. Now, in this figure, you can see here the hematopoietic stem cells arising in the bone marrow differentiate into immunocompetent T and B cells in the primary lymphoid tissue. Then, these cells colonize the secondary lymphoid tissues where immune response occurs. The mucosa-associated lymphoid tissue mount together with diffuse collection of cells in the lamina propria produce antibody for mucosal secretions. Now, the location of the immune system. Let us discuss this. Virtually, all cells, exception being the follicular dendritic cells, of the immune system are generated from multipotent hematopoietic stem cells in bone marrow and majority of these cells mature within bone marrow prior to being released into the blood circulation and subsequently entering the tissues. Now, the cells of innate response are found throughout the body. As resident, for example, resident tissue macrophages and mast cells in connective and mucosal tissue, like, for example, NK cells in many locations. For example, neutrophils also recruited to the site of infection, while lymphocytes of adaptive immune response, although B cells become fully mature within bone marrow, as we mentioned, T cell precursor must travel from bone marrow to the thymus to be fully mature. In that case, where the bone marrow and thymus, primary lymphoid tissue, as we mentioned, in which mature lymphocytes are produced. Now, any location in the body outside the primary lymphoid tissue is called periphery. Just keep in your mind that any location in the body outside the primary lymphoid tissue called periphery. Initiation of adaptive immune response by lymphocyte occurs in specialized area of the periphery called secondary lymphoid tissue, mouth, lymph node, and spleen, as we mentioned previously. Now, how does one categorize a cell as belonging to the immune system? It's a good question. In fact, there's no easy answer to this question. For example, why not? We can see that RBCs are not usually considered a part of immune system, despite the fact that these RBCs have receptors for complement, which provide them with an important role in clearance of immune complexes from the circulation. So why not? Why we can't say the RBCs are part of immune cells? Also, but it's not immune cells, in fact. Also, an endothelial cell are also not classed as cell of immune system, while their role in altering leukocyte into an infection site. Now, let us discuss something very important related to the humoral antibodies, immune response, sorry. There are types of B cell that respond to an infection, to an antigen, I mean. Follicular B cells, we call B2. These cells present in lymphoid follicles of spleen and lymph node require T cells in order to generate high affinity antibody and to undergo class switching. Also, we have another type of B cells, innate-like B cells. Innate-like B cells such as B1 and marginal zone B cell, MZ B cell. There are certain types of antigen called T-independent antigen can promote B cell activation without the help of T cells. As a result, antibodies formed have low affinity in that case to this antigen and do not undergo class switching or somatic hypermutation, but in turn they provide rapid protection from certain pathogens in that case. until T-dependent B cell responses formed or occur. Innate-like B cells express tool-like receptors. That's the reason why we call it innate-like B cells, and can be activated by PAMPs, pathogen-associated molecular patterns of the pathogen or antigen or any foreign substance, act as antigen-presenting cell and secrete cytokine, and that puts us an interface between the innate and adaptive immune response. For example, in the skin, mucosa, and in marginal zone of spleen, where the lymphatic and circulatory system converge. Now, B cells can respond to 3 different types of antigens. What are these types of antigens? First, type 1 thymus-independent antigens. Certain antigens, such as bacterial lipopolysaccharide, when present at a sufficient high concentration, have the ability to activate polyclonal B cell pool without reference to the antigen specificity of the surface receptor hypervariable regions. That means binding through tool-like receptors. In that case, the tool-like receptors provide the binding site for these antigens. Second, type 2 thymus-independent antigens. Certain linear antigens that are not degraded in the body and have spaced, highly repeating determinant like pneumococcus, polysaccharide, for example, and many substances like glycol, D-amino acid polymers, all these able to stimulate B cells directly without the need for T cell involvement in that case. Such antigens persist for a long period on the surface of follicular dendritic cells located at the subcapsular sinus of the lymph node and splenic marginal zone, and can bind also to antigen-specific B cells with great avidity through their multivalent attachment to the complementary immunoglobulin receptors. We are going to discuss multivalent, univalent in a lecture of antigen-antibody interaction. In general, the thymus-independent antigen gives rise to low affinity IgM responses, and if there is memory, it will be very poor in that case. Let us mention some very important note. In neonatal B cells do not respond well to type 2 antigens, and this has important consequences for the efficacy of carbohydrate vaccine in young children. In young children, they use carbohydrate vaccines in that case because of this reason. The efficacy of B cell in that case drive this response. Also, another note, the marginal zone B cells located in the marginal zone of spleen and monitor circulating pumps. B1 B cells found in the skin and mucosal surfaces, as we mentioned, and act as rapid first line of defense against microbial invasion. Now, important note, activating both these innate B cell types will promote a strong IgM and IgG3 response and also present antigen to T cells, leading to activation of adaptive immune response. Now, the third type of antigens is thymus-dependent antigens. Here, there is need for T helper cells, collaboration in that case. These antigens cannot provide direct stimulation because they may be univalent, or also they may be degraded by phagocytic cells in that case. And if such antigens bind to B cell receptors, they just bind on the surface just like a hapten and can trigger B cell activation. As we mentioned in immune response lectures, hapten cannot induce the immune response unless it binds with a carrier molecule, larger molecule, protein molecule. In that stimulation, hapten cannot stimulate antibody production. That means it cannot stimulate B cell activation. As you already knew that both T and B cells are necessary for antibody responses to thymus-dependent antigens, so here there is need for T helper cells stimulation in order to cooperate with B cells for further response to hapten by providing accessory signals in that case. Now in this figure, you can note that while one determinant on the atypical protein antigen is acting as hapten in binding with B cell, the other determinants subserve a carrier function in recruiting T helper cells in that case. Now, as I mentioned in a previous lecture, that B cell sometimes act as an antigen presenting cell. Now, how B cell act as antigen presenting cell? T helper must, as we know, that T helper must recognize the carrier determinants on the responding B cell in order to provide accessory stimulatory signals in that case. As T cell only recognize processed membrane-bound antigen in association with MHC molecule, T helper, as you know, cannot recognize native antigen bound simply to the immunoglobulin receptors. So this antigen need to be processed into peptide and bounded to MHC molecule in order to be recognized by T cell. In that case, B cell in lower concentration of antigen work as an antigen presenting cell. It can present antigen to helper cell, but in different way as antigen presenting cell is the conventional antigen presenting cell, because B cell can focus antigen on through their surface receptors. Now, what are the steps of antigen processing by B cells? First, antigen bound to surface immunoglobulin and then internalize in endosomes to enter inside the cell. Then these endosomes fuse with vesicles containing MHC class II inside the B cell. Processing of the protein antigen then occur into small peptide, the resulting antigenic peptide, then recycled to the surface in association with the MHC class II molecule, where it is now available for recognition by specific T helper cell. As you can see in these figures, figure 725 and 730. Also, from these two figures, after helper T cell recognize the antigenic peptide in combination with MHC class II on the surface of B cell, it will be stimulated and then stimulate B cell to differentiate, proliferate, and produce antibodies. These antibodies can react with the same antigen that presented to T cell. In the second figure, 730, you can see that T helper can either recognize antigenic peptide complex with MHC class II on antigen presenting cell, like dendritic cell for example, or on B cell. Now let us discuss what is going on in the lymphoid germinal center. In order to understand the clonal selection, the clonal expansion, diversification of and affinity maturation of B cells, all these processes occur in germinal centers to generate high affinity antibodies. These antibodies play a critical role in humoral immunity then. In germinal center, control process of rapid B cell evolution occurs. How this occurs? This occurs through maturation of their B cell receptors. Follicular T cell helper cell select the highest affinity B cells on the basis of their ability to capture antigen from follicular dendritic cells. In that case, in this figure, in the absence of antigen, B cells within lymph node reside within primary follicles, in that case composed of a mesh of follicular dendritic cells. As arresting small B lymphocytes upon encounter with T-dependent antigen, then the primary follicles transform into secondary follicles. As you can see from this figure, it's very clear how the transformation occurs. Now, here antigen-specific B cells undergo a rapid clonal proliferation and crowd around follicular dendritic cell network, displacing resting B cell into a periphery or mantle of the follicle. The mantle zone, I mean, in that case. Now, you can see the details in the figure that there are two anatomically distinct regions. The regions is the dark zone in the above side of the figure, and then the light zone. The dark zone contains large mitotically active B cells called centroplasts, while light zone below. Light zone contains smaller non-dividing B cells called centrocytes. Also, as you can see, the antigen deposited on the surface of follicular dendritic cells, like a dot on the follicular cells. And there are also antigen-specific follicular T helper cells, which are critical for generation and maintenance of follicles, of course. Here, B cells with high affinity is selected. That means only high affinity B cells are selected in this zone, and then after migration of these cells between the two zones, the dark and light zones, this process occurs continuously. In dark zone, location of B cells, proliferation and maturation, and selection provided by T follicular helper cells. Now, also B cells might then return to dark zone. First, after migration to light zone, the B cell then return to dark zone for multiple rounds of proliferation and maturation. During B cell division in dark zone, somatic hypermutation occurs. In B cell, immunoglobulin genes might occur in that case. Also, B cell undergo immunoglobulin class switching also in this region. Now let us discuss something about how when B cell, if B cells are rescued by T follicular helper T cell. What I mean by that? I mean that choose the high T follicular T cell, choose the B cell that are high affinity to antigen in that case. In light zone are protected then. The B cells are protected then from apoptosis, while unprotected B cells, that means with low affinity of antigen, will die readily and taken up by macrophages. Macrophages will take these cells with low affinity to antigen and apoptosis and to get rid from these apoptotic cells. In marginal zone center, the B cells can capture antigen by their BCR receptors, then present it to the germinal center resident T follicular cells as antigen presenting cell. After that, T follicular helper cell can influence B cell selection. Here, B cell will be selected in that case at this step. As a summary for clonal selection of B cell clonal expansion is controlled by a limited number of T follicular helper cells. In light zone, these T helper follicular cells give help to B cell which captured and internalized the most antigen with high affinity. It's very important to be high affinity for antigen for the B cell to be selected. The more antigen captured by B cell and presented on MHC class 2 to T follicular helper cell, the more likely it undergo further proliferation and somatic hypermutation. I mean for the B cell. Now let me ask a question. What is the reason for the separation of B cells into light and dark zone with, with the germinal center? Why there are separation between these two layers, zones, I mean? The answer for that, in order to minimize the antigen depletion in that case, because the antigen will be depleted and the whole process will be confused in that case. Now let us discuss something about BDJ recombination and diversity. First of all, you have to know something about the human immunoglobulin loci in order to understand BDJ recombination and diversity. As you can see here in this figure, the immunoglobulin heavy chain is present on the genes coding for immunoglobulin heavy chain is present on chromosome 14. The BH variable F heavy chain, nearly 40 functional gene. The diversity D diversity heavy chain, it's 23 functional genes, and the J, the junction heavy chain, nearly 6 functional genes. The lights here mean the leader, the L, I mean in the figure you can see at the start L1, L2, until L40. The L here means the leader sequence. For immunoglobulin lambda chain, the genes present on chromosome number 22 contain the variable, the lambda 30 functional gene and the junctional lambda 5 functional genes. Each J segment followed by constant segment. As you can see here, C lambda after each junctional segment. That means C lambda 1, C lambda 2, C lambda 3, C lambda 4, and C lambda 5. Okay. In the immunoglobulin kappa chain, the genes coding for immunoglobulin kappa chain present on chromosome number 2. We have variable kappa, 40 functional gene, and junctional kappa, 5 gene functional genes. Now, let us discuss the steps of BDJ recombination and combinatorial diversity. In the figure 322, in this figure you can see how BDJ recombination occurs. BDJ recombination is highly regulated and ordered event. The L light chain exon is constructed from a single B to J gene segment join. At the heavy chain locus, D segment is first joined to a J segment, then B segment joined to the combined DJ. First of all, as we said, the D segment joined to J, and then the D joined to the DJ. The rearranged DNA then transcribed into RNA, messenger RNA, and the RNA transcripts spliced to make V region exon and C region exon together. Then the spliced messenger RNA translated in order to produce the final immunoglobulin protein. In this way, numerous unique immunoglobulin genes can be made by joining different combinations of BDJ segments at the heavy and light chain loci. The diversity in the immunoglobulins through this joining called combinational diversity. Also, additional diversity is created by pairing of different heavy chain with different, for example, lambda light chain or kappa light chain. Okay. Now, let me mention something about recombination signal sequences, the RSS, in figure 323. The RSS is non-coding sequence that flank coding gene segments. It helps to guide recombination between appropriate gene segments. The RSS, the recombination signal sequences, is made up of a conserved heptamer and nonamer sequences separated by an unconserved 12 or 23 nucleotide spacer between these segments. Efficient recombination occurs between segments with 12 nucleotide spacer and 23 nucleotide spacer. That's mean 12, the ratio, the rule of 12 to 23 rule, help in that certain appropriate gene segments are joined together in that case. In variable heavy locus, the variable and junctional segments, the V and J segments flanked by RSS with a 23 nucleotide spacer. As this figure shows that how it's happened. The D segment, the diversity segments, are flanked by RSS with a 12 nucleotide spacer. The light chain loci, variable kappa segment, flanked by RSS with 12 nucleotide spacer, junctional kappa, JK, with 23 nucleotide spacer. This rearrangement is reserved in the locus of lambda locus in that case. Now the recombinase machinery. How this recombination occur? Which enzyme playing a part in that case? The BDJ recombinase is a complex enzyme that mediates somatic recombination of immunoglobulin gene segments. Like in this figure 324, The gene product or products of recombination activating gene 1 and 2, RAG1 and RAG2. RAG means the recombinase enzyme. Recombinase activating gene 1 and recombinase activating gene 2, RAG1 and RAG2, are lymphocyte-specific enzymes essential for BDJ recombination. Now, the class switching recombination, the CSR. As we mentioned previously that antigen-stimulated IgM expressing B cells in germinal center of secondary lymphoid organs, the spleen and lymph nodes, undergo the class switching recombination, the CSR. The CSR allows immunoglobulin heavy constant region exon of an antibody, for example, to be exchanged for an alternative exon, giving rise to expression of antibodies with the same antigen specificity but different isotype. For example, the IgM will be class switched to IgG, IgM to IgE, IgM to IgA. All these occur regarding to the type of infection, type of antigen, type of immune response required. For example, as we can see in this figure, constant region exon for IgD and IgG, IgE, and IgA isotypes are located downstream of IgM. This is in the germline, and there are we can see the mu constant region exon and CSR. The switch recombination occur between the switch region, the S region, Here you can see in the figure S mu, S gamma 3, S gamma 1, S gamma 2b, S gamma 2a, and so on. S epsilon, S alpha. Here, the S regions are repetitive sequences. They are guanine-rich sequences located on the non-template strand and found upstream of each constant heavy chain exon, as it's clear from the figure in that case, except in C delta. In C delta, you cannot find the S region in that case. Now, how the class switching recombination occur? By making breaks in the DNA, into the DNA of two S regions. Between two S regions and fusion of S regions leading to a rearranged constant heavy chain locus. In that case, a variable exon is joined to an exon for a new constant region. Now, the DNA between the two switch regions exists, cut, and form a circle in that case. As it's very clear circle here from the cuts of DNA, and then binding the two regions with each other in order to form the messenger RNA for coding the new immunoglobulin isotype. Finally, alternative splicing of primary RNA transcript generated from the rearranged DNA gives rise to either membrane bound or secreted form of immunoglobulins in that case. Now, as a final step here in this figure, now the expected immunoglobulin that will be synthesized according to the messenger RNA. Here, IgG2A messenger RNA will be synthesized as immunoglobulin G2A, as it's very clear here from according to the messenger RNA created in this case as a result of recombination. Because the cut here made in the DNA from S mu to S gamma 2A. Okay, and all segment in between, as we mentioned, cut in and make a circle. And messenger RNA now know what is the code he that should be synthesized, and then code for the IgG, as we mentioned, IgG2A instead of IgM. Okay. Thank you very much. Any further question, we can discuss it through the Telegram or free conference or any way. We can discuss through the lecture time. Thank you.